Just going out on the airways, doing it. Yes. Is mine up someplace? Because I've had requests for it.
It should be on YouTube. I think, Bobby put it up.
Okay, all right. Which one?
The one that didn't do it.
Okay. I was no longer a host. Each time I joined with my friends to be a co-host.
We don't even have audio for it. Is the line? I thought we did it.
I'm still waiting to go in the party. Okay.
You sure?
Recording in progress.
Basically, sound hitting. You know, just. This one. Well, a microphone. Holy. I don't think I've ever worn a microphone. Hold on. Guess that. They have some new. the microphone work at all?
Yeah, it's muted.
You're looking at my butt. I know it's okay, but yeah, it's unmuted. All right. I can hear, yeah, it's on there. Yeah, so it's not out here, but that's fine. So it's not in the room. It's on there. You're gonna have to turn me down because I'm gonna end up talking loudly then. All right.
If it doesn't start when we got starting, we just split this. And
Yeah, we should be good.
it should be good.
Yeah, I appreciate it. Thanks, man.
Okay, so.
Nobody likes that side of the room? How you doing? Brent?
Steve Midway.
All right, nice to meet you.
He's our fish guy, one of our fish guys. Yeah.
The only two ologies I didn't take were micro and ick.
Well, fishology.
Yeah, fish,
And
didn't
I
do,
was telling
didn't,
him about our conversation.
Yeah.
And he said that he would be interested in possibly developing a fish course for all.
okay, Yeah.
We have undergraduate fish courses that need some new life in them, to be honest.
yeah, yeah. So are you, now, I'm guessing, you're not in this department, or are you in this department?
yeah.
Okay. You guys have ick in other departments too, don't you?
Yeah, we're split.
Not really?
Sort of freshwater ick.
Okay,
And we've got icks and herbs. And we have more.
Yeah.
And Evo ick that's in biology.
Yeah.
The fishery stuff is here.
well, so, and that aspect would be really good, because again, I can't teach ick at all. I mean, literally, you don't want me to go down that path whatsoever. And so, yeah, and so just being able to have students who are interested,
Yeah.
right, to be able to give them that option, it allows me to multiply the possibilities for my students beyond me. And I tell them all the time, you're limited by me right now, so you've got to find other ways to learn. Teaching yourself ick is going to be a challenge, but having some support, that'd be great.
Yeah, I've taught it before, I've taught it under ichthyology with more of a sort of ecology, what does this
Yeah.
matter in the environment kind of?
Yeah,
Because, counting fin rays and stuff is nice, but a lot of times the students, they want to be able to sort of
Why do they care?
understand the why
Yeah.
as to not just the what.
that's especially true for my students here. Your students have, seminars and workshops and diversity of courses. Most of my students will take ecology and maybe a 100 level kind of environmental science seminar.
Mhm.
And beyond that, there is no kind of applied aspect to what they learn. The challenge is going to be, it'll probably be a mixed class of grads and undergrads,
Yeah,
which I don't
we do
care.
that here
Yeah, I
too,
don't care.
so,
Yeah. We're not supposed to do it, but we all do it all the time.
Yeah,
You might
yeah.
mention that, yeah, you're not supposed to because it's fact,
Yeah.
but, you might mention where your students went. You had what, 7 students?
Yeah, I spend half my time talking about my failures and in like trying to overcome those. And one of those for me is the success of my students. So we'll talk about that.
Okay, good.
Yeah,
Great,
Yeah.
I look forward
So,
to it.
all
Thank
right.
you so much.
no problem.
Yes.
Is there, I'm gonna go grab a bottle of water real quickly. Actually, I'm gonna mute myself and I'm gonna And let me be perfectly clear, I don't do climate change research. And I was involved in this all of 1 month or about a month and a half before I came down here. So there's not that we're starting the process of building the relationships. So thank you all for coming and listening to me. It's been great to be here. I'm at a university that, despite being an R2 and trying to build towards R1, has very limited research in marine and environmental sciences. And the fact is, I am the marine science program. We technically have a marine science program. I was hired to be the director of the program. And then I found out that I am the program. That's problematic. We have an environmental science PhD program, used to have an environmental science master's. And actually, we have a concentration in environmental science and marine science at the undergraduate level. The challenge is 20 years ago, late 90s, early 2000s, these were really dynamic programs at Jackson State. About 50 environmental science students, 25 marine science students at the undergraduate level. We had about a dozen faculty working across that, a lot of interdisciplinary stuff that was going on. But luckily, I think LSU, and some of you have been here long enough to be able to correct me if I'm wrong, but is large enough to have enough momentum to overcome changes in administration. Jackson State's not that way. We had a change in department chair who saw the need for more biomedical science and slowly suffocated new hires in those fields. So when I started at Jackson State in 2016, again, hi, marine science, the entire program. We have one person who's now doing phytoremediation, and all of the rest of the environmental science is actually environmental health. So to be able to come and talk with you all about anything related to the ocean, the coast, the environment, it's like a breath of fresh air, right? Because otherwise you're sitting there in your office going, I'll go talk to my student who knows, can't really stimulate the conversation quite the same way. So it's been great. I want to thank you all for having me and allowing me to talk with you all. And I look forward to continuing to kind of develop relationships. We've had a number of conversations about potential collaborations and things we can do and helping to move the DEI needle when it comes to the ocean. Remember, about 1.5% of ocean scientists are Black. That's almost criminal, 1 to 1.5%. So anything we can do to change that is a big thing. Now, we got all that good stuff out of the way. I want to start with the weirdest beginning of a talk probably ever. I'm sorry. I'm sorry. This is not a normal talk by any stretch of the human imagination. Instead, I want to tell you a story about my time trying to breathe life, CPR, bring marine science back at Jackson State. The challenges, many of which are my own, the lessons I've learned. And here's the deal. A lot of you, I think, are graduate students. Some of you might be postdocs. Don't make the same mistakes I made. Now, your advisors are telling you already not to make these mistakes. I'm an outside guy who you don't know from Adam, but trust me, don't make my mistakes. And I'll tell you what they are and how they've impacted where I'm at. I'll also tell you about how we've made real progress in changing things. I think it's important to understand that we face challenges not just in DEI, but at HBCUs and other MSIs with how we can change things, okay? So it's not a real research talk. Yes, we'll talk a little bit of science in there. We'll talk about some of the projects we're doing, but I want to tell you a story about, again, kind of the progress, the challenges, and the successes that we've had. So I'm sorry. If it's boring, I apologize. Now, my background, I'm from Missouri. I grew up loving outdoors, but as I was telling somebody earlier, when I was young, I thought if you wanted to work outdoors with nature, you could be a vet or you could be a conservation agent. The only reason I knew about conservation agents is because my uncle happened to work for Missouri Department of Conservation. I'm a middle-class Midwestern guy, fairly educated, and that's what I thought you could do, right? And so when I had the chance to go to the Gulf Coast Research Lab in Ocean Springs, Mississippi as a sophomore, I was a rising sophomore. There's a lot of backstory there involving dropping out and going back to school, but not necessary. I had a chance to take a marine invertebrate zoology course. And as part of that course, I met a couple of gentlemen, John Foster and Richard Hurd. Richard Hurd was an older gentleman. At that time, he was in his early 70s and still was going out in the field showing. I remember one day I went up there to his office and I was trying to identify 2 different crabs, and he was literally wearing dress slacks. It turns out he was dating somebody he ended up marrying for later, whatever. Dress slacks, dress shoes, tie. And he's like, oh, sure, I'll go show you. He's walking out in the marsh in dress shoes and digging up crabs to show me, this nobody from Missouri, these different crabs. And I remember how inspiring that was, right? And so by the time I graduated with my bachelor's degree, literally the night before my last final, Organic 2, in case you're curious, I submitted my 3rd manuscript for publication as an undergrad, because I went back the next couple of years to do research experiences. And here's the thing, research experiences for students change their pathway. It changes their lives. It makes the difference. It's such a huge deal. Went back there, did my master's, did some more stuff, but I love systematics and taxonomy. I loved finding these things. I was collecting Pokemon before Pokemon was cool, right? And that was the thing that I loved about it. So after my master's at GCRL over in Ocean Springs, went to the University of Louisiana Lafayette, and I kept collecting Pokemon, doing phylogenetics and systematics taxonomy of xanthoid crabs. So for those of you who work in the estuary, you've seen Panopeas before. These are a lot of the mud crabs, Eurypanopeus on the oysters, Panopeus up in the low salinity marshes. And I was doing phylogenetics, systematics, and taxonomy of this group. This is a phylogeny from a zillion years ago when I was a PhD student, presented this somewhere, and mainly just to have something up here to talk about. This type of work was fun to me. I was unraveling the mysteries of how these organisms are related and trying to figure out, what's new and what's not. I loved it, but one thing I didn't do, and this was mistake number one. Again, we're telling the stories of my saga of sorrows so that you don't make the same mistakes. I didn't look to the future. I looked at what I enjoyed, collecting Pokemon, although I didn't know Pokemon was really a thing until I have kids of my own now. But and I never really thought about how I'm going to fund this research. And I kind of blame my advisors because they were having fun doing it, and they were at the point in their career where they could keep doing what they wanted to do and didn't have to worry about it. But I never stopped to look and say, how am I going to fund my research down the road? How is this going to keep going? So that was mistake number one. Finishing up my PhD, doing a postdoc, started looking for jobs. And knew I didn't want a high pressure R1 research intense thing. I didn't want that pressure. Turns out I got it anyway. But I started looking for jobs, and I started interviewing at Jackson State, Mississippi. And that was second, third year. I turned down some other positions because I'm an idiot. Again, bird in the hand's better than two in the bush. Remember that. It's there for a reason. When I got there, during the negotiations, the dean says to me, we don't do startup funds. We do core facilities. We invest in core facilities and core labs. We believe in building synergistic activities, and so we invest in core facilities. Okay, that makes sense. You're a smaller school. I can understand that. He offered me $2,000 startup and access to the core facility. Hubris, right? Messed me up. I thought, well, I'm good. I can make that work, right? I can make that work. I accepted the position. Okay, so welcome to Jackson State. I come to JSU in January of 2016 with this idea that I'm going to breathe life back into marine science. I'm going to change everything. I mean, in 5 years, there's going to be more African-American marine scientists walking this planet than ever before because I'm going to do it. I didn't. It's not true, not at all. But that's what I felt like. And so I went there and I said, no startup, no problem. Now, when I was touring, I was shown a number of potential lab spaces. There was this beautifully remodeled lab space. It was an absolute dream, right? But I didn't nail it down. Instead, when I got there, I walked into a lab that looked like this. It was literally an old teaching lab that they've been using to dump their surplus and their junk into for the better part of a decade. My first year and a half of my research was spent removing literally hundreds of gallons of waste, cleaning stuff out, disposing of things, and cleaning up. I started in January 2016, did not get this done until March of 2017. Okay, that's how long it took me, because I'm also teaching a full course load. I don't have any students. It's me and like work study students. Mistake number two, well, number three, the startup will come back to be a mistake, was not figuring out where I was going to be. All right, so always look to the future. No matter how much you're doing right now, look to the future. So in 2017, I am faced with no real funds for research, a lab space where the totality of my equipment included an old microscope that I found. It was actually had a camera on it. It was all right. Luckily, I am a geek, a super geek. And so when I was an undergrad doing those papers, I bought a microscope from a retiring scientist so I could do my own scientific illustrations when I went back to Missouri. So I had my own microscope and I had the school's microscope, and that was it. My chair came to me in that summer and said, hey, we've got a program. We're bringing in students to do research in non-biomedical science programs. Oh, that's awesome. And he goes, since you're pretty much the only one doing that, we have 7 students that want to work in your lab. Oh, there's $3,000 in supplies. That's not going to work well. But all the travel you want. I like traveling. I like going to the beach. And so with 7 undergraduates with varying backgrounds, most of them are pre-professional in concentration. One was marine science, one was an environmental science concentration. I tried to come up with a study that did really 2 things: keep a bunch of students busy, that's important, and get a bunch of students exposed to marine science in a fun way. Again, I'm in Mississippi, you guys are in Louisiana. You know, marshes aren't necessarily the most fun habitat in the world for people who are not really into them. So every month, my plan, brilliant plan, every month I was going to take students from Jackson, Mississippi to Navarre Beach, Florida. It's a beautiful beach. If you've never been, go sometime. Beautiful, so white sand beach, calm, very little wave energy. And the plan was to take this group of students every month for 3 years. Why? Because I could rotate through and add new students in and think of all the students I could expose and potentially drown. That comes back too. We started going in September of 2017, loaded up in the van, drove down there. First trip, van broke down. Not a big deal. We fixed it on the side of the road. It wasn't a big leaky hose, no big deal. Got out there to start collecting. And when we got ready to collect, again, beautiful beach. Our plan was to collect at the swash zone, so right here in this area, and then about 10 meters out. You don't want to go by depth because literally it could vary laterally very quickly. So roughly about a meter deep. Go collect, both these depths, 3 transects, 3 replicas each, and collect them in this gigantic core. It's literally about that big around, push it down, fills up a 5-gallon bucket. And I know that because we were going to sieve them through a 0.5 millimeter sieve, pretty standard for doing macrobenthic work. Challenge is, this sand is not like marsh sand. It's not like marsh sediment at all. It doesn't go through a 0.5 millimeter sieve. First trip with a broken down van, we drove to Ace and I bought every 5-gallon bucket they had in order to get the samples back to Jackson State. Things are still going good. I got 7 students going with me. 7 students came back. They were all alive. There were no major injuries. We go back in October and the waves are much bigger. The seasons are changing and things have changed. So we're out trying to work out in this zone with 3-foot waves. At that point, I nearly drowned a student. That's not an exaggeration. Aspirated water, come up coughing. Turns out she couldn't swim. She got knocked down, panicked. We broke one of the cores, lost some other stuff. And I said, maybe I shouldn't be doing this 1 meter depth all the way through, but we'll ride it out. We went back in November, broke another core, and the students refused to go out and work in that depth. So we're 3 months into the sampling study and we have to get rid of this sample site. And at that point, I realized I'm not publishing this, right? You've realized the whole plan is falling apart. Over the next 13 months, and I'm going to put this up here just so you have something pretty to look at, right? Don't worry about the science. I wouldn't trust it, but it's there. Over the next 13 months, I went to Navarre Beach every single month, 1 week in a month, and I took students with me. And over the period of those 13 months, I took 27 different students with me, built around a core of 7 that went all the way through, 2 more that joined later on. And those 7 went back and worked in the lab. We were averaging somewhere around 80 hours a week between these students working in the lab. And upon graduation of those 7 students, every single one went to a postgraduate program of some kind. Every single one. Now, remember, I'm at a historically Black college where less than 5% of our students in the biology department go on to professional programs, maybe 10% total in some type of postgraduate program, and I hit 100%. Out of those, only 2, kind of 3 were biomedical science. One's a nurse practitioner, one's in his 4th year of med school now, right? I have one who's in a PhD program at Princeton working with African savanna nutrient cycles. He's living the dream. How many biologists want to go work in Africa and watch elephants and wildebeest poo? Okay, maybe not the poo part, but that's what he does. I have another one who's working on a PhD in entomology in UC Riverside studying bees. Ironically, she'd never been stung before. Turns out she's allergic. You might want to check into that before you get into bees. Another one, this is actually one of the ones that came on in the 2nd year that just got to go in the last 2 sampling or 3 sampling periods. She's now doing a master's in oceanography at URI up in Rhode Island, which is one of the best oceanography programs in the country. Master's in biology and master's in physiology. But anyway, I also had another one of the students that came in, went and got a master's in environmental science at FAMU. I have another one that just started the program and a former student who's now in the 3rd year of his PhD in environmental science at Florida A&M University, by the way. We got to figure out how to get that to be LSU instead of FAMU. We can talk about that. Oh, here's what we found, just for the record. basically in fall, when the water temperatures are starting to drop, we see a great recruitment. Emerita donax, which is the conchina clam, same kind of trend. I don't really accept this. If you know much about these organisms, they're really, really patchy. They're really, really patchy. And I literally probably could have moved 3 feet down the beach and got a totally different set of data, but I kept 7 students busy for $3,000 for a year and a half, and ultimately 2 years, and I got all of them into something else. So yay, I did it. The only problem is I still have $2,000 startup. Technically, I used it to buy a microscope so we could actually do this work more efficiently. So I'm broke. I still have no publishable research. I still have no funded grants, and I'm now in the 3rd year of my time at Jackson State. Now, faculty know mid-tenure review. That's a thing. I didn't have that. That's a whole other saga we can go into. Importance of knowing what you're supposed to be doing, even if you're not told. But I started to panic, and the reality is I gave up. I was telling somebody this morning that something inside of me, even I didn't recognize at the time, said, wait a minute, if I can't do research, because again, no startup, no equipment. Oh, I didn't get to the no equipment part, did I? I had $2,000 startup. So I get my lab cleaned up. I go to the core facility and I go to the director of the core facility, Dr. Stevens, and I say, hey, I'd really like to get trained in what I need to do to be able to use this facility. And she looked at me and goes, you can't. But the dean, I'm sorry, it's only for people funded by RCMI, which is an NIH funded grant, or doing biomedical science research. We have a number of certificates and things in place that prevent us from allowing other people to use the lab. But the dean, and I went to the dean, he goes, I wasn't aware of that. That was the end of the conversation. That was the end of the conversation. So here I am again. That actually happened in 2018, 2017. I was trying to do some mitogenomics with some of the animals we got out here on the beach when I was showing students other stuff. And so at this point, I'd given up. I'm going to do the best I can for the time I have and ride it out and give up. Luckily, I have a wife who loves me and wouldn't let that happen entirely. And fortunately or unfortunately, COVID happened. Right? So during COVID, I went from serving students. I have an open door policy. I created a student organization focused on getting students into RUs because, again, they changed my life, and I know they'll change their lives. We went from, in the summer of 2015, before I showed up, we went from 2 students out of a class of over 700 doing REUs to last year, it was 32 or 34, I can't remember, students doing REUs. Still a miserable, miserable percentage. 600 plus students, and fewer than 40 of them did summer internships. Miserable percentage. So I spent all my time working with students and building students, and then COVID hits and I'm stuck at home. And at first I panicked. What am I going to do? Who am I? I can't help students if I'm at home. But I started writing proposals, really writing proposals, not the one a year that I threw as a token at NSF in order to try to, check that box on the theoretical mid-tenure review that never happened. Instead, I started really focusing on writing every proposal I could. And luckily I found somebody, a collaborator I never would imagine, a gentleman from Sudan who's been alive longer than I've considered, who's done every job possible, has a PhD in physics, a PhD in electrical engineering, set 20 years apart, worked as a postdoc in a biophysics lab doing really weird stuff to cats and their nervous system for 4 years, traveled the world. And I ran into him because he called me, he goes, hey, do you know anything about oysters? I've got money to work on oysters. Well, no, I can eat them, cook them. I know where they live. I know what lives with them, but I don't really study oysters. He goes, well, you do now. Okay. I have no money. You've got money. Cool. I'll do whatever. We started writing proposals together. In January of 2020, I had $0 in grant funding. And again, was considering what I'm going to do, probably paint houses when I lost my job. And by January of 2020, by January of 2022, $3.2 million in collaborative funding, right? And it's not me, I'm not that good. Most of it's training money. And what I learned was, hey, you can get money to train students a lot easier than you can get money to fund research. But if you do it right, you can use that money to build the program you need to build. So we're still in COVID, and I'm getting this stuff. I'm writing proposals. We wrote 7 proposals in less than 12 months. The first proposal I get was a NOAA proposal, and it was basically focused on doing the same kind of thing that we did here, getting students out in the field, getting some minimal training, and trying to build that pipeline. The problem was Jackson State would not allow us to travel with students until November of 2021. So I think it was September, or I think it was September 1st of 2020, I was given a couple $100,000 to do this project over the next 5 years. And I went to NOAA and I said, hey, I can't do what I said because they won't let me travel. It's COVID's fault, not me. Don't blame me. But can I please take some of this money and revise the project to focus on lab skills that would be useful? And they let me spend $120,000 to build a beautiful name, Environmental Genomics Laboratory. It sounds so good. It's a closet that I cleaned out and put my equipment in and bought new stuff, but it sounds great on a grant proposal. So it's the Environmental Genomics Lab, $120,000. And finally, I could do the research I was trained to do and more. All right. Keep proposing. We get students in there. And I just want to kind of go over where we're at and what we're doing. That's the story of how we got to where we're at. Things have changed so much because of making one connection, right? Making one connection. Kamal reached out and saved me. My wife wouldn't let me give up, and having support around you is incredibly important, whether you're a graduate student or whether you're a faculty or whether you're a street sweeper. Having people to support you is incredibly important, and don't give up. Perseverance. How many of you have had advisors tell you to keep trying, persevere, you can do it? Have you gotten to that point yet? If you haven't, your graduate career is much different than mine. I considered quitting every single semester, and that's not an exaggeration. Every semester I looked at, maybe I should quit and go do, I always went back to painting houses. I make good money painting houses. So my work now is mostly focused on environmental DNA. Why? Because it's a tool you see being used for a huge number of different projects, right? And whether we're talking about metagenomics or metabarcoding, whether we're looking at I don't think Jenna's in here, viruses or bacteria, whether we're looking at eukaryotes or fishes, environmental DNA is something that's of interest to NOAA, to USGS, to ONR. It's of interest to a lot of people. And it's something that uses the same skills I already had. So I started looking into this, just for those of you who don't know, because I know we have a fairly diverse background. Environmental DNA is basically a DNA suspended in water, soil, air, whatever the case may be, in this case, water. It could come from microbial cells, virus cells, metabolic waste, feces. We'll talk more about poo later. Urine, could be tissue, blood tissue, mucus, anything like that, skin, scales, hair, whatever's suspended in the water column, right? Free DNA from ruptured cells. We can collect that by filtering the water, extract the DNA from that, and then we have a couple of options. If we're really interested in one species or a narrowly defined group, we can develop probes for those groups and look at quantitative approaches to estimate, detect them, estimate abundance. Harmful algal blooms, we can detect them very quickly at very low levels. Or if we want to understand the entire community, right? I want to know what lives in this area because I want to see how it changes over time. I can use next generation sequencing approaches for metabarcoding, and I can basically make a list of everything that's there. Using different markers, I can look at microbes, or I can look at kind of general eukaryotes, including largely protists. I can look at metazoans, or I can look at fish. I can use RBC for phytoplankton or ITS for fungi. And so you really have a lot of power with this approach. And training, right? My goal is to get my students that when they leave, they have a STEM career, whether it goes on to graduate school somewhere else or whether they get their master's or their bachelor's with me and go work as a technician. This requires them to learn a lot of laboratory techniques, some bioinformatics skills. Every single one of my students presents. It's a whole thing. It's all built around helping them have skills that they can use. And hopefully the confidence, it's a big deal, by the way, amongst my students, the confidence to think they can go on and do something more, right? And so this is the direction that I was moving in. It's changed even more so in the last 6 months. But one of the first projects that came across that was really funded for research. Now, we're not just training, but research is a project working with U.S. Fish and Wildlife. They do a lot of work with crayfish. We'll talk more about that in a second. And they wanted a way to work with crayfish, detect crayfish, identify crayfish without having to dig up some farmer's field. Turns out they don't appreciate that. All right. Just if you weren't sure, they don't like their field dug up. And so we started talking about using eDNA for this approach. We'll talk a little bit about that study. And then again, going back to that NOAA training proposal and some of the work we're doing with it, I realized one big thing. Field work is important. It's important to getting students interested in marine science. I think most of us like being outside and on the beach or on a boat way more than we like being in a lab, moving a pipette or back and forth. But I did not want to go to Florida in January or February ever again if I could avoid it. Okay. So we started doing projects looking at using fecal DNA. So we literally capture fecal pellets, do metabarcoding with that in combination with environmental DNA, and try to understand diet and trophic ecology in marine invertebrates, particularly those same marine invertebrates that are in that sandy beach in Navarra. Because again, I'm trying to hook students on marine science. You don't want to take them to Grand Isle if you're trying to sell them on marine science. No offense to Grand Isleans in here. So when we think of crayfish, most of us think of something like this. I lived in Lafayette, Louisiana for 10 years, and a special place in my heart for that picture right there, right? But the reality is, this is what you should be thinking of, right? A lot of crayfish are stream crayfish. They're what they call primary burrowers. There are ones like this that live near wetlands where the water table is maybe a few inches below the layer of the sediment there. And they have these large burrow mounds that they've produced by excavating these huge burrows that can be 3 or 4 feet underground, down to where the water table's at, so they can stay. You got gills, man. Got to keep those gills wet. And so they're digging down to where the water table's at. So again, you're U.S. Fish and Wildlife, and you just had a list of 900 species petitioned by, I think it was Nature Conservancy. Anyway, a huge list of petitioned species, and it included every species of crayfish in the state of Mississippi was petitioned for adding to the Endangered Species Act, which is pretty hilarious. But anyway, so they're out doing research, and I apologize for some of the blurry pictures here. So you can be this lucky dude from the U.S. Forest Service who's digging down past his shoulder into the mud, trying to find the crayfish, digging down there, hoping you can get it. Or you can be Xanthia here, who's working with a slurp gun. If there's a water table high enough, you can literally slurp them up out of their burrows. Problem is, some of these live in areas where the water table's 2 or 3 feet down and the burrow's 3 or 4 feet down to get down to that water table. So they're trying traps. Here's a trap door trap. It crawls out to go out and mate. They trap it in the top part of this tube and they come out and get it. But every single one of these has major drawbacks. First off, like I said, farmers don't like that. Nobody wants you digging up their yard. It takes a lot of work. It's a lot of effort. Maybe if it's the right crayfish and it's in the water table. Otherwise, again, you're coming back out multiple times. You're trying to get these things and it may or may not work. And so they wanted an approach that would allow them to determine, when they look at these burrows, which crayfish is it? Not have to dig them up, not have to collect the crayfish, which crayfish is it? And so we proposed the idea of using eDNA to understand basically the pattern of distribution and habitat use in Procambarus barbager. This is the Jackson Prairie crayfish, cool looking dude, looks like it's got pom-poms, yay, go team. And this is the historic range. You can see the red records are the ones after 1990. I love the way this is worded, and before 2000. So if it was collected on New Year's Eve, it's lost. We don't have any record of it. But basically, you can see here we have a number of records here off of Jackson. Jackson is about, I think it's right there, but whatever. Get the idea. And then we have a few records that were collected more recently. Now, that's a fairly broad range in terms of some of these endemic crayfish, but it doesn't make sense. When we actually look at it, this is the Jackson Prairie crayfish, and it turns out it's known from the Jackson Prairie ecotone, a specialized ecotone with unique soils, unique plants. It's its own thing. Never knew it existed. Lived in Jackson for 6 years before I ever heard of the place, knew it was an actual thing. And so when you look at it, these make sense. What's going on here, right? And so the goal now is to go out and work and collect from throughout the Jackson Prairie ecotone, as well as the entire drainage basin they think they should be in. And every time we find a little burrow, we're collecting the burrow. Not the whole thing, that's too much work. We basically are collecting the mud. And the fun part is it has to happen right after it rains. And if you're not familiar, it rains down here a lot in January, February, March, sometimes April, which means we're out playing. We really start the week after next for our main field season for these. We'll go out and collect them right after a heavy rain when the burrows are fresh. You can see how this is still fairly wet mud, not dry like the top of this one is. They form these by using the mud in their mouth parts. They literally kind of roll them up in balls and deposit them on top, which means they have lots of DNA on them. We go out and collect them. We were originally collecting them. When we started working to prove this would work with U.S. Fish and Wildlife, they basically would go out with a plastic bag, turn it inside out like you'd use to pick up your dog's mess. And they would scoop up the burrow wall, turn it inside, zip it up, eventually right on the outside. And then sometime along the way, they'd throw a paper tag in there, which is really good for keeping other people's DNA out of the bag. But we've learned since then, we actually take a beautiful microcore, also known as a stainless steel straw, in case you were curious and want to buy one at Walmart. We go out and we collect a very small sample. We literally will shove it down just right on the edge of the burrow where it's moist and has the maximum traffic, take a microcore, preserve that microcore, and from there we're extracting the DNA from them. Inhibitors are a pain. If you've never worked with environmental samples, PCR inhibitors are a real pain in the rear end. It makes it really hard to get amplification. These are not our worst samples, but it makes the cost higher than we planned, but it's still doable. The nice thing is we're able to get good DNA from these. We're able to get good eukaryotic DNA with CO1. We're able to show that there are crayfish there. The downside is, and this goes back to my love of Pokemon, inverts, and taxonomy. We don't have barcode data for most of this stuff. If you're not familiar with DNA barcode, despite the fact these are often considered threatened and they're petitioned, there's a huge amount of crayfish that we simply don't have good barcode data for. So I can tell you that that's a crayfish burrow, which by the way, you could have told from looking at it, but without having the barcode data to match it to, I can't tell you which one it is. So now we're faced with a challenge. That wasn't part of the original plan because we were assured by our colleagues in Fish and Wildlife and Forest Service that there was plenty of that stuff out there. They would hook me up with, that was actually the words, hook me up with the people who had the data. They don't have the data. So now we're having to build the database of Mississippi crayfish to do this work. We're starting out with metabarcoding. We want to find out everything that's there. We want an idea if there's stuff living in the burrow. It turns out there'll be clams in some of these. There'll be mice that use parts of the burrow. And so we want to understand the community, a little added benefit. The goal, once we get the data set for those metabarcodes, develop TACMAN probes and use quantitative digital PCR, because this cost me $100 to $120 per sample. This cost me $20, $25 per sample, and that's a lot cheaper, right? And so we have a student that started on this project in September. That's when the project started in October. So I started paying her in September off other money. She started on this in October. And so far, like I said, we've proven that we can get the DNA. We've proven this is a better method. Field season starts next month. Yay. I love rainy, cold mornings. That's one of the projects. And I love this one. I love this one for two reasons. I don't work on crayfish. I don't work on crayfish. I never worked on crayfish in my life. I eat crayfish. I've never worked on crayfish. It turns out I work on them now because it's a way to take my non-applied phylogenetics and taxonomic understanding, the stuff I love to do, and do something else with it. I finally looked ahead, right? I finally looked to the future. Secondly, U.S. Fish and Wildlife is another one of those agencies that's serious about DEI. And they have a number of programs for opening pathways for students working in these areas. Already, we went from this being a trial to 2 internships this summer with U.S. Fish and Wildlife that are joint shared between me and Fish and Wildlife. They'll be working with them and with me. Another pathway to get students into another field where they're woefully underrepresented, right? All right. Fecal DNA, diet analysis, and all of that. This is not an invertebrate, in case you were curious. Just want to get that out there, but it's a good picture to understand stomach content analysis and what we do. Pull out the stomach. We got a bunch of stomach and stomach contents. We tease these out and separate them out, and it turns into a beautiful chart like this. This is not my work. This is just simply to show you what traditionally happens. We get a list of the genus of the CDs. We get some Penaids. Other crustaceans can be fairly. Crab, that's really useful. It ate a crab. We don't know which one, but it ate a crab. A fish, a gamerid. And this is what we traditionally have. And it's fairly labor intensive, right? You have to collect the animals, you pull out the stomach contents, you're looking at them under a microscope, and we can see not much of anything from that, right? That's literally a pile of mush. And we come through, and again, I want you to tell me which species of squid this comes from, right? It's like you're finding the beak in a part of the fin, or what did this come from? And the challenge is, that's why we end up grouping, when we're looking at stomach content analysis or diet analysis, we end up grouping them very broadly. Crab. So using the same eDNA approaches that we are using with crayfish and that we're trying to develop in the lab, we want to understand diet, but in groups that doing even this is impractical. If you, for example, are working on a ghost shrimp like this one, this is Lepidophthalmus louisianensis, the estuarine ghost shrimp, pretty common in this part of the world, judging by the name. Louisianensis. We think it's a filter feeder, and I'll get back to that in a second. So if you're a filter feeder, how am I telling what you're eating? It's all mush in your stomach already. How am I telling what you're actually feeding on? And so we go out and we collect them. This is my PhD student, Julian Venable. This is me with my hair even longer, right after COVID. Yeah, rocking the man bun. It was beautiful. Collecting the ghost shrimp. This is in right behind Pensacola and Navarro Beach. There's a nice site where it's actually really pretty again. You keep going to pretty sites if you can. We collect them. Here's the group working on it. We put the shrimp alive in filtered seawater. We wait until they defecate. We collect the food pellets, fecal pellets. They're actually little pellets. They kind of look like something you would feed a turtle or your fish in your aquarium. They're fairly robust. They don't break apart super easily. We pull them out of here. We preserve them in DNA shield. We then preserve the animal because there's some reasons for that in a moment. And we can come back to the lab and we end up with really 4 types of samples. One is the fecal DNA pellet. Here you can see the fecal pellets. This is actually from a mole crab and not from the Lepidophthalmus, but just a nice, beautiful picture. Usually mole crabs have much smaller, ghost shrimp much larger. We take an environmental DNA sample from the water. We're filtering 1 to 2 liters of water using a Sterivex filter. This is a fairly common approach for eukaryotic metabarcoding. We then also collect burrow walls. And what's cool about this is this species, Lepidophthalmus, live in fairly organic, rich sediments, low salinity, kind of estuarine, again, estuarine ghost shrimp habitats. And unlike most of the ghost shrimp, they have a distinct burrow wall. It's very organic, rich. It literally, we call it the turd tube. It literally looks like a piece of fecal matter that's been hollowed out in the middle, dark, rich, sometimes smells, and it's kind of unique to them. And the reason I mention that is because we believe, based upon some of the cetacean of the mouth parts, that they're filter feeders. That's the long-standing tradition, but there's some observations in the aquaria of these grazing on that wall, and not just grazing on the burrow wall, but then taking fresh sand and their spit, their digestive, and they pack it back on there. And there's quite a bit of again, suggestion that they may in fact be farming fungus and bacteria in that burrow wall and then grazing on their own farm bacteria and fungi. So the idea is this: if the community that we see, the genetic community composition that we see in the fecal DNA better matches that we see in the water, they're probably filter feeders, and I wasted the time doing all this. Oh well. If it better matches the burrow wall, then they're definitely feeding on what's in the burrow wall. All right. The other problem is when you're doing any of this, guess who's in the burrow and guess who made the fecal matter? This guy. So we have to start by developing blocker primers. We have to block out their DNA. So now I got an undergraduate student who's involved working in this project who's helping with that aspect, learning DNA extractions and Sanger sequencing and techniques that for a lot of us aren't current techniques, still valuable. As an undergrad, they get to get those experiences. Remember, the idea is train students, and the research is great. I would love to publish it. I need to publish more. But my goal is to get well-trained students out so they can go somewhere else. So now we're doing it at 2 levels. This is one of the examples. Because I'm traveling to the beach, I'm not going to collect one animal. We're doing another ghost shrimp that actually lives on the other side of the sound. So this is in the estuarine side. That's the back side of the island. There's a different species of ghost shrimp called Calichirus halagrande, lives in the high energy side, right? And that's actually a really cool story about trophic dynamics. And so we're actually comparing their diet on both sides of the island, on both sides of the sound. In addition, I have like 5 more minutes, right? I got to pick this thing up then. You say that, we could be here all day. We're also doing Emerita, the one I showed you earlier, the mole crab. It's a filter feeder. The question is it a selective filter feeder? And again, these are getting students out in the field. We went out 3 times last summer, collected at different locations, learned about proper techniques for record keeping, preservation. We also learned some really cool things. Turns out that even filter feeders don't feed at night. They're dormant. If you go out and collect these animals that are thought to be filter feeders first thing in the morning, they will not defecate. You can put them in a tube and leave them there until you grow old. They will not defecate. If you go out 3 hours later and collect another set, within 5-10 minutes, all right? It's even more impressive with the predators that we were looking at. We're looking at some predatory mole crabs that are good carnivores, scurry around and eat everything. You have to wait till 2 or 3 o'clock in the afternoon before they have eaten enough to defecate again. So go late at night and avoid sharks. That would be my lesson. All right. Like I mentioned, Kamal, my buddy in engineering, he reached out to me. One of the first projects that we collaborated on was working with these sensors they developed using Hall effect sensors for monitoring oyster gate behavior. I want to talk about that because it's a cool thing. And It's kind of one of the directions that my lab is going, whether I want it to or not. Basically, little Hall effect sensor, a Hall sensor, a magnet on the other side, and effectively by measuring the voltage change, you can measure the distance between these two. So as the oyster or the mussel or clam opens the shells, the distance between them changes, the voltage actually drops, and as a result, you can measure the gape distance. Now, not just the gape, but also as they grow, because the baseline, when they close, will change as the shells grow. It's used extensively in a lot of shellfish research, particularly in Europe. It's now quite common on the East Coast of the US when people are doing aquaculture and laboratory stuff, measuring these. But the problem is it's limited to the lab. You'll find huge tanks with big apparatus hanging over top of them. What we've done that's different is this is field deployable. We can literally tuck this inside of a bag of oysters, go out and throw it in the ocean, come back and recover it later on and have real world data on what they actually do. That's cool. But what's even cooler is that when we look at the real data, this is an actual data. You can see here the gape readings. This is how large the gape opening is. It's a somewhat artificial number, right? Remember, this is based upon voltage and not actual distance. But we can see how here it's wide open and periodically it closes and closes. We don't know what causes it. A lot of times it's a startle response. It's sometimes a way to help clear out water if they've got something that's fouling them up, clogging their gills. But notice this pattern right in here that looks like something crazy is happening. That's a really cool aspect of what we're doing. If we look at it, I'm gonna play a video and I know it's time to get out of here. Uh-oh, I killed something. All right. So you can watch the valves here slowly coming back apart. It's opening back up. And if you watch, they will actually pulse close very, very rapidly. And so when they're spawning and they're pushing those gametes out into the water, they'll close rapidly and you can really see a cloud of gametes actually come out of them as they're doing it. Turns out that's what this is. So we went to MDMR over in Mississippi and talked about the possibility of being able, I don't want to restart that. Being able to use this in the field to detect spawning behavior, which of course then helps us determine where the spat are going to end up and look at larval retention and things like that. So they said, that would be amazing. Can you get us real-time data? So this is what we do to try to figure out how to separate that noise, right? How do we pick this out automatically from this so I don't have to watch, I don't have to have students looking at, bags or samples and samples and samples of this. We can have something pick it out automatically using neural networks. And so basically we realized if we do a fast Fourier transform and we average the results across certain number of readings. So this is 100 readings, that's 10 seconds. So every 10th of a second, it goes back 10 seconds, averages the gape distance. And when we do that, if we start to look at about 1,000 readings, so 100 seconds, you can see a clear spike. And it turns out we can teach a computer to look for that in the data so that we don't have to look at the data at all. And with that, we can automatically detect spawning behavior, okay? Now, the problem is we want real-time data. And so we're currently integrating cellular data. And I say we, I'm not my engineering buddy, right? But he takes credit for my work. I'm taking credit for his work. We are doing this. And so basically a lander that goes underwater, it's got all our processors and everything else, a little cellular radio. He's too used to working cheap. I keep telling him we can afford to buy a better cellular radio. Has a little fishing float. We wanted to stay as small as possible. This is literally a little wooden cork because we've deployed 3 different versions of earlier things of these that would just to collect real world data. And every single one was not there when we went back 2 weeks later. It happens. If you've ever deployed gear, you've left it somewhere, you come back and it's sometimes not there. So a little tiny float sensor. This is another cool device we'll talk about some other time. The challenge is that antenna has to be above water. And it turns out waves with an antenna and cellular signals interrupt it. You have data transmissions that don't go through, it cancels them out. Number 2, it takes a ton of power to make a phone call every 2 minutes. You only get about a 3-day operating window. So now we're having to change the transmission frequency. The problem is if you lose one of those transmission frequencies and the handshake doesn't happen, you lose data. So we're having onboard storage, right? Super cool. Looks like we're getting ready to get a renewal of funding for this to actually get this implemented and get real world testing. COVID, of course, hurt that as well. Part of the problem with this is as we look at trying to mount the sensors on spat and smaller oysters, our techniques don't work. Hot glue on a big oyster, not a problem, doesn't mind a bit. Turns out hot glue on a spat turns into a cooked spat. We tried a UV cured glue. If you've ever seen those little Bondex commercials, if you stay up way too late at night and have your TV on, there's that UV, the little pink or purple light you use. That works beautifully. It holds the sensor on. Unfortunately, it also kills the oysters. Apparently, it has some toxic chemical. Keep that in mind if you ever use it on something that you want for something else. There's some chemical in it that leached into the oysters and killed every single one of them in about 2 days. I laugh. It's not funny, but it's kind of funny. The problem is we're doing this research at the Gulf Coast Research Lab over in Ocean Springs, 3-hour drive from Jackson. We drive down, we set it up, we get it all set up there, and we drive back and we come back, and the next day, all the oyster gapes are wide open because they're dead. And then Jackson's backwards in a lot of ways. Jackson State's backwards in a lot of ways. It takes me about 2 weeks to get travel approval if I'm asking for no funds. I was joking with somebody yesterday. I literally got approval for my travel for here yesterday while I was having lunch, and I submitted it back in January. Okay. And so this is a month-long process to redo this. And so we've got to figure out a way to get wet labs at JSU. And so as part of our grant writing frenzy over the last little bit, we got money to kind of set up and again, I love titles, right? Big naming titles sounds great on proposals. The Aquaculture and Aquarium Science Lab at JSU. We got some money, it's not much. And we're starting small. It's a 900 square foot facility. Again, sounds impressive. It's an old teaching classroom in another building that I paid to have everything ripped out of, paid to cut out the floor and put in drains. We're going to start with 6 closed loop mesocosms. We control everything individually on those. And there's lots of room for expansion. We literally could put in about 3 times that much in this room. And so it's a huge step, again, as to our capabilities. And again, it's progress, right? Challenges in progress. This helps us move forward. There's problems though. We started in September of 2021. We got permission to do this. I had the money in place. We started the demo in November of 2021. By the way, here's the funny thing. I got permission after I got the money. Turns out that was smart. Then they didn't ask me how I was going to pay for it. Phase 1, we had to have everything, the floors cut out, literally come in and put drains in. If you're not used to working with tanks, they always leak. Something always breaks. There will always be a mess on the floor. Plan ahead. So we cut out and put drains in. This took way longer than it should have. Supply chain issues. Contractor, I think, went on vacation for 6 months and blamed supply chain issues for the drains not being put in. We waited for quite a while. We finally got those put in late September. We started phase 2 in October. Retractable overhead, blah, blah, blah. Lots of cool stuff. This also hit by supply chain issues. I think he didn't order them. He claims he ordered them in early November, but these retractable overhead cords, again, cords running across the floor plus salt water, bad plan, unless you don't like your students. And so these were back ordered. They were actually putting them in when I left on Wednesday. I went down and checked on it Wednesday because I wanted to be able to say something about this. And they were literally putting in, they had 5 of them in, we're putting the rest in there. So maybe when I go back, we'll be ready for phase 3, which is actually starting to put in the real infrastructure, right? The part that I have to do, unfortunately. And so this is exciting for me because it's not just for these experiments. We have students that are wanting to do work with microplastic exposure in oysters and how that impacts their gene expression. I've already had students approach me about starting an aquarium club at Jackson State, which, by the way, anything that gets students doing something that involves salt water, I'm down. And heck, I'll even do fresh water. I'm fine. As long as I'm doing something that's not biomedical. So this is exciting for me. Yeah, that gets me in trouble a lot saying stuff like that. But this is exciting for me. This collaboration with Kamal and his group over in engineering, and I know I'm past time, I apologize, is important because one of the things it does is allow me to try new stuff. If you're familiar with eDNA, it requires you to be there at the time you're going to sample it. Whether you're using an old school, this is actually for bleeding the brakes on your car. People are using it to create a vacuum and they're filtering out in the field. Cool backpack things that take 3 samples at one time. Maybe you're filtering bags of water you collected, or even you're on a boat and you're collecting massive numbers of samples, but you have to be there when you want to collect the sample. So how do you do time series analysis, right? How do you look at how things change in short scale? So Luke Thompson, his group in NOAA developed what he calls a SASE. It samples using 2 pumps, takes 1 sample, and he can program it to take that 1 sample whenever he wants. So he'll go throw it out in the beautiful coral reef. Wish I worked in the coral reef. And it collects a sample at some time period, middle of the night or whatever. But if he wants to collect more than 1 sample, he's literally got to daisy chain these things. He throws them out there and he has a whole set of them, 8 or 10 of them on the seafloor where he can dive down and find them. I live in the Northern Gulf. If I throw an individual thing down there like this and there's no buoy on it, I will not ever find that device again, right? Now, this is not the only one that's out there. Mbari, working with USGS, have got one about the size of a 55-gallon drum. It's a large, big ocean deployable one. They use this one on the side of the stream. It's an impressive apparatus, but it costs huge amounts of money, custom engineering. Same thing, if we look at this is one you can buy from Dartmouth Ocean Technologies. It collects, I think it's 12 samples, it's 8 samples, 10, somewhere 10 to 8, whatever, some of that area. And it has proprietary filters, proprietary equipment, all that kind of stuff. This is Imbari, and of course, Imbari's got everything. This is a custom filter. One of these contains preservative, one contains a filter, one contains a gaseous nitrogen that purges the preservative afterwards. And it's a custom little tiny thing. You can see a bunch of them on here, and it goes to their $1 million drone and collects samples. Now, again, I work at Jackson State University. Nothing about all this custom is going to work for me. And so we have been prototyping a automated eDNA sampler that will collect up to 100 samples. This is basically a grid of these individual little filters. Here you can see a Steravex filter that we use. These are used quite commonly by NOAA for a lot of their eukaryotic eDNA work. It'll sample 100 of them, basically uses an XYZ actuator. These are sealed motors. We're going to have custom machine. Probably, that's my prediction. We're trying stainless steel first, but I'm guessing custom machine Delron. Delron is a really hard plastic. That way it won't corrode. Turns out salt water is bad for metal. And so that's part of it. Electronics, pumps, tanks, batteries, that's all engineering stuff. You don't really care about that. The cool thing is 100 samples, minimum 30 days operation, and that's determined by the preservative effectiveness. Nobody knows how long these samples can be sustained in DNA shield out in real world temperatures. We always preserve them, bring them back and throw them in the freezer until we get to them. How long can we actually keep the DNA? That's our real limit there. Batteries somewhat, but you can add more batteries. He doesn't like it when I say that, by the way. Apparently you don't tell an engineer you can add more batteries. Apparently that's a bad thing to do. Onboard sensors, salinity, temperature, pH, DO, and turbidity. And so while we're collecting the samples, we're not just collecting the samples, but we're also collecting the environmental parameters so we can try to understand what may be causing the changes in community composition as we're looking at this. And there's, I want to mention this because, you know, if you're deploying gear, this is actually something really cool that the engineer, Kamal and his team have come up with, called underwater time release. No more buoy sitting on the surface. We deploy the gear, and at a predetermined time, it triggers the buoy, releases it, it pops up. So if you know you're going to go collect it on this day, your buoy shows up at that day. Nobody's stealing your gear, nobody messing with your gear. They're trying to integrate acoustics so that you can trigger it acoustically so that you don't even have to pre-program it. If you need to do an emergency release, you can. That'll be integrated. I push that one because that's one that they actually have that I think a lot of people should be using when they're deploying gear. All right. All of this is ongoing. I'm done with sand beach invertebrates to go out and look at temporal things. Monthly sampling is for the birds. It's a lot of fun to get students out there, but again, just December through March is not as much fun as it sounds like. eDNA is a big part of it, whether it be looking at this and integrating students into these pipelines, whether it be engineering students who are doing their senior design projects and are tasked with doing certain parts of this, or whether it be undergraduates we fund off of GeoPass funds that are helping in the lab, is a big part of what we do. Again, not a normal talk, right? I think I presented one really bad slide of really bad results that were literally produced by an undergraduate like 3 years ago. But I hope it helps you understand how things are progressing, like the challenges, but also the fact that we've done a lot, and maybe helps us figure out a way that we can work together to increase that 7 to 70, right? The 7 students to 70 students. Acknowledgements, I'm not going to read them all. You guys have eyes and can read. So I'll answer questions. I do want to point out again, Kamal and his team, crucial to the last 2 projects, and of course, the funding that we've been working with. And with that, 10 minutes late, I'll take questions. And that's how you kill questions, go 10 minutes late. Somebody's always obligated to ask a question. Yeah, go ahead. Hold on, back. Are you talking about the oyster behavior or which one? So here's the challenge. We have yet to do any in the lab I'm responsible for. Every time, just touch the mic. You don't do that. Everything we did was deploying it on Gulf Coast Research Lab. And here's the thing. They don't keep track of any of the parameters. They literally come in and feed them, and then they walk out and there's no change. We don't have any evidence of what's causing the behavior. So that's part of why when we build this lab at JSU, the one big thing we asked for funding for was to actually try to understand what that gait behavior reflects, right? And so that's why we can control all those different parameters individually for each mesocosm, so we could try to interpret it. Just glancing at the data and talking to them, there's some other figures I didn't show. It's crazy how much light apparently influences their behavior. I did not think about that, but they would have things where we're pretty sure the light came on in the morning, the oyster stayed closed the entire day, light went off at 5 o'clock, oysters opened back up and fed all night, which is crazy because they're actually fed during the day, right? But they were staying closed all day long. And we think it's tied to either the action around them, the disturbance, or that light, which doesn't make sense because, you know, there's a sun up there, but it is what it is. So we don't know, to answer your question. Yeah. We have not, and should have. The reason I'd be hesitant is the time for a silicone to cure is fairly long. My suggestion, and they keep fighting me on this, and I don't know why, that's why when the lab I'm responsible for, we'll use extra thick super glue. If you've ever done any kind of aquarium stuff, you can glue corals directly to stubs and all that kind of stuff with super glue. Heck, if you cut your finger open, you can glue your own finger back shut with super glue. By the way, don't use regular super glue. It burns really badly. I heard that from a friend. But yeah, so we hadn't talked about silicone, extra thick super glue, because you can even use an accelerator on it and cure it almost instantly. The problem is it also produces heat. So accelerator probably wouldn't work. Yeah. Yeah, Marine Text is one of them. They also use the red and white one that's by, yeah. All those are fun things. And again, you got a student, you got an undergraduate student who can test them on different ones and let me know what's going to happen. I love those kind of things, right? So, all right. Yeah, yeah. If you haven't noticed yet, I like to talk and I'm quite good at it. I've learned at a young age to talk. I'm the son of a preacher also, and so I'm good at selling things. The student organization did was a big part of it, right? So I was hosting regularly. I'm talking about twice a month, CV workshops, personal statement workshops, how to find an internship, how to write a research statement, how to get a good letter of recommendation. I have cut way back on that post-COVID because again, the research side has picked up, and that's an unfortunate thing. And there are repercussions of that for this coming cycle, but it is the world we live in. Number two, with that student organization, we had over 250 people on our group me from our department. So I would post regularly to the group me stuff. Number three, for a short period of time, I had money for a program manager to help me out with some of this stuff, and her background in marketing and education, I was trying to leverage that. She did really good on some and really bad on others. Social media, but here's the thing, I'm old. And unless I can get somebody who's young, who's willing to do it, we try to use students for social media, but if there's problems with that, right? And so all of that, right? It's a lot of things. And I also teach one of the core courses that every biology student has to take. So everybody has to get through me at some point. And I'm beating those REUs like nobody's business. I mean, I will come in and I will put them up. I'll have a series of them flashing up that I've got in my inbox at night before I start my lecture. They'll be rotating through that on a separate slideshow. I will stop and talk about them. Luckily, they'll stop in about 2 weeks or so. When we get to the 1st of March, I kind of stop. If you haven't applied by then, I'm sorry. But yeah, it's a lot of things, and I wish it were more. It turns out no matter what I do, I can't make them apply. And that's the ultimate end of it all. Yeah. Not that I'm aware of. Not that I'm aware of. It's used again a lot it was originally a European thing. They were using them for their offshore mussel farms that you have out there dangling down. They were doing them in tanks. I have not read anything about its influence on them. It might. My only thought is that they're not moving. They're not going to be using magnetism for anything that it might influence, but it could. That's a good question. And so I didn't talk about some of the AI work. I work a lot with the engineering and the computer science group now because that's my collaborators apparently. Like I said, I have nobody else to talk to and nobody else to work with, so you find new friends. And so we're doing some stuff with AI and neural networks and fish scales. And so a lot of what I do is I get students to look at stuff, and we could actually use basically a combination of cameras and AI to compare the behavior of gait in censored ones and non-censored ones. Because again, nobody's going to reliably watch tons of video footage of oysters slowly opening and closing. So yeah, that'd also be a cool project. Thank you. You maybe give work for somebody else. I like that.
Final questions? Great. Well, let's thank our speaker again. I'm going to lunch in the